Okay, thanks for hanging out for the whole time. I suppose if there was a break, a lot of people would have left. But anyway, I'll I'll crank through this last one. So I want to recognize a lot of co-authors. I was a lead on a study that capitalized on a lot of data that Lummi Nation had collected, and I was invited along with Corey Green of NOAA to help analyze that data and collect a couple more years of that data. Basically, I never work in the Nooksack Basin or Bellingham Bay. I work out of Skagit in the Whidbey Basin. I do live in Linden, but what they wanted was us to help fill a data gap that was identified in the recovery plan for WIRA-1 Chinook or WIRA-1 recovery plan, and specifically for the Nooksack Chinook. So we were attempting to sort of repeat analyses that were done in the Skagit and look at sort of fill in sort of the natural history of the juvenile Chinook through the estuary and nearshore environment. I also want to acknowledge, I was asked by Alan in particular to kind of connect to the Salish Sea Marine Survival Project, a couple of studies. So both these guys, Josh and Lance, I know and are friends with, and they gave me some slides and some material to help connect. Although, while Michael said I might talk about some of these things in detail, I only had 15 minutes and Michael had 30 or more. So why this study? We're filling the data gap. I'm going to blow through slides because I see one lesson I learned today is that people were running out of time. So the data gap was identified in a recovery plan, and this study filled it. We had funders, the Bellingham Bay Action Team and City of Bellingham and DOE were primary funders also, although Long Live the Kings helped out too. We didn't do anything cool. We did basic bootstrap type of stuff. You go out and beach seine things. I have some pictures of beach seining going in the slides, but we didn't do any sexy modeling or GIS really, just simple stuff. So we capitalized on over 50 sites from Lummi Nation that they had been collecting data over the last about a decade, and then we had 2 years of data collection where we sort of built on that record and added some spatial extent and balance to the design so we could fill this data gap of juvenile Chinook use in the estuary as well as Bellingham Bay. I don't have time to go through very many methods, and I'm pretty dissatisfied with having for some results being just telling you the answer rather than showing you through data, through results, but multi questions. One of the main ones, and the basic one, is just sort of identifying the population structure of the juvenile salmon. So we use terms of natural origin recruits and hatchery origin recruits. So we're looking at where they're living and when they're living in these basic habitat areas. And then it includes what size they are and absolutely the relative abundance. I'm not going to show any size data today, but it's inferred when I talk about the life history types. And so it's already been mentioned, things I think Michael had talked about fry and par life history types. And in general, life history types are within the Chinook population and any salmonid population, you have juveniles living a different way to complete their life cycle. And that, by having that diversity, you end up with a portfolio effect. You don't have all your eggs in one basket. So you end up with a more resilient population if you have these diverse life history types. So part of our study was to look at that. And I'm going to belabor the concept a little bit and show you. This is like my only conceptual slide. So what we have is 3 different parts of the system. The blue polygon is the freshwater environment. The tidal delta, the natal estuary, is the yellow polygon, and then we have the nearshore system in the pink polygon, Bellingham Bay. And so in each of those places, there can be branching. I call it phenotypic branching. We think that the fish are adapted to the unique river. There's genetic work that shows that in general across the geography of Chinook salmon range. But within a river, we have local adaptation, and that may be somewhat genetically controlled or genotypic related, but within the river, we have these phenotypic expressions. So that what the fish experiences in this environment, how many fish are around or what habitats are present or absent, can result in branching. Every fish emerges, if it survives, the eggs survive, it emerges as a fry, and it has a chance to stay in fresh water for a period of time, sometimes months, sometimes over a year, or migrating downstream. When it reaches the estuary, it has the same sort of thing. It could migrate through quickly, or it could delay and rear for a while. And the same thing when it gets to the nearshore system, it could go into what I call unique, these refuge habitats. They're sort of unique places. They're sheltered from wave energy and predators and those sorts of things. So pocket estuaries and even some non-natal streams are these sort of refuge habitats, and some don't find those habitats. So there's branching in each of these boxes, and that's the phenotypic expression. And so when you have fish leaving the Bellingham Bay nearshore system and moving offshore into the Salish Sea, there could be 5 different life history types. So we're looking to see if those are present in our system as we did in the Skagit. So keep that in mind as I go through with some of the words. So first we capitalized on the Lummi Nation's extensive main stem trap data, smolt downstream migrant trap. They run a rotary screw trap near Hovander Park and make population estimates, and they fraction out the population estimates by the fry, par, and yearling life history types. And what that data shows is that all those life history types exiting the lower river are present. On average, there's about 190,000 juvenile Chinook leaving the system each year. Of course, it widely varies, and it is dominated by the par migrant type. And there's a whole section in the report, and I don't have time to talk about it, but there was analyses done that looked at whether there's evidence of density dependence or that juvenile rearing habitat in the freshwater system, whether it's filled up, and it was quite compelling that that habitat is not filling up. It's not anywhere near capacity. And there's recommendations about how to think about that. And Ned and others had talked a little bit about some of that. There's also hatchery origin fish in the system. So they could, when you're looking at our study system, we have to include nearby other waters like the Samish River or Lummi Bay, which is actually part of the Nooksack estuary, historic estuary. But there's, on average, about 5 million hatchery fish released into the system. So a couple 100,000 natural origin fish and 5 million hatchery fish. That's what we had to deal with when we go out with our beach seine and potentially catch fish. And fortunately, marking practices are really good in this basin, actually, and across Puget Sound now. It didn't used to be that way back in the 1990s, but basically you can you have very low risk of erroneously catching a hatchery fish and thinking it's a natural origin fish. So this is kind of the nuts and bolts of it. So where were the natural origin and hatchery origin fish living in the Nooksack Tidal Delta and Bellingham Bay shorelines? And kind of the bottom line is that we found consistent use of the Nooksack Delta by the natural origin fish, but the densities were lower than in Bellingham Bay. That's kind of surprising to me coming from the Skagit River. It's the opposite there. And then within Bellingham Bay, there was consistent use by the Norfish in the pocket estuary. So the places that are like Paddon Lagoon or Squalicum Estuary, and even Whatcom Waterway and I&J Waterway, those were defined as pocket estuary-like because they have freshwater inputs and they're really sheltered from long wave energy and those sorts of things. They're sort of artificial pocket estuaries. And then if you remember, I said there was a couple 100,000 outmigrants on average of the Norfish and 5 million in the system of the hatchery fish, but we caught an order of magnitude more of the natural origin fish than the hatchery fish, in spite of the several orders of magnitude difference of release in the system. And that implied that those natural origin fish are really delaying in those rearing habitats, those shallow, complex tidal habitats in the estuary, as well as in the pocket estuaries in those areas. Just some graphs to show so what the data looks like. Just the 2 years that we had good spatial representation, or better spatial representation. So 2014 on the top, 2015 on the bottom, different out-migration years. Blue bars are the natural origin fish, and on the x-axis it's months, and y-axis it's fish density, and they're all the same x-axis, so or y-axis and x-axis. But lots of Norfish early in the year in the Bellingham Bay shoreline. Hatchery fish showing up in May. That's good because that's when they were released, and lower densities in the delta. Within Bellingham Bay nearshore, this picture divides it into two kind of habitat types. Whoops! Oh, those are bullets coming on. Sorry. Within Bellingham Bay, we divided it into two habitat types. So the exposed nearshore habitats, so not the pocket estuaries, they're exposed to wave energy, and then the pocket estuary habitat type like places. So that the y-axis is quite different. They're an order of magnitude different. So there's many, many more fish in these pocket estuaries compared to the more exposed nearshore areas. And then we had one small stream out on Portage Island that we were able to sample, and it sort of shows the same pattern of high densities of fish early in the season, like late winter. Fish going into those as refuge habitats. So our study, we didn't measure ecological interactions, but we did document, or made a point of documenting when you had commingling, and mainly within the pocket estuaries and the natal estuary, we don't find for early season, the fry period, we don't find commingling with hatchery and natural origin fish. We find the commingling only occurring in the more exposed nearshore habitats later in the year. The hatchery fish, of course, don't get there till later in the year. There's some other interesting things about the study. We looked at connectivity of habitats, and basically within the delta, not every place is easily accessible to fish. And there's a bunch of things about that. Parts of it anthropogenic, in that we have road systems and dike systems that are kind of making habitats hard to get to for some places. But there's also natural things going on, like the channel spanning jams that formed during our study period, which put water and sediment in different places in the delta. And that was interesting, a good experiment to look at our fish data and see whether there was effect on that. And basically there was post-log jam. It deflected water to the west and fish to the west, and some of the sites on the east side of the delta had less fish, and those habitats resized. So those are interesting things to think about when you're thinking about restoring the habitats. You might want to restore connectivity, account for strategic places where fish are being delivered to in your restoration sites. So we highlighted that in the report too, and just sort of give you an idea of the broad spatial bins in our polygons. Basically, this is what we call the connected tidal delta, and that's where most of the fish are living. It's logical because the river is going directly into that. But the Silver Slough area is fairly close to the river too, and it has very, very few Chinook living in it. And distant but historic, estuary is the with remnants of habitat there is the Lummi Bay area, and that had very few fish. And just sort of a simple bar chart of that is shown that all the fish are in that one area, and so that leaves some room for restoration of connectivity in the system, and maybe some things to think about for the Silver Slough area too. Briefly, we capitalized on the Marine Survival Project to do some lab work and looked at origins. That's important because in the marine area you can have mixing. And so we looked at that, and kind of the bottom line is in the estuary you have Nooksack populations dominated by the early timing in the early part of the spring populations in the early timing. Within the pocket estuaries, there was also Nooksack origin fish, including the spring populations, but as well as the naturalized falls. It wasn't until you got in the Bellingham Bay nearshore habitats that you started seeing the out-of-system fish. In particular, you see Whidbey Basin fish getting up into Bellingham Bay kind of late in summer. A big thing of the study was whether there was density dependence, and I think you probably know the answer. There's not a lot of fish in the system to outmigrate, so we didn't see density dependence. The estuary is not at carrying capacity, but it is being utilized by these different life history types. We had to look at growth, and this is where growing is good for survival, and this is where I was asked to try to link it to this the marine survival study. So we found that functional conditions exist within the estuary and in the nearshore environment. We inferred it functional because we looked at fish diets and we looked at temperatures and we modeled growth. So there's food there for salmon and there's good temperatures. We had 2 years of data in multiple habitat types, and we found that no single place was like the optimal place. In 1 year, it was good to be in a pocket estuary, and another year it was better to be in the delta. Those are differences in temperature, and it kind of goes back to the life history type diversity. You probably want to have some diversity to kind of offset the risks of things being different in year-to-year basis. So with that, we tried to, Josh and I tried to link, or did link, his offshore data from the Salish Sea survival with our data. And sort of the original hypothesis of the Salish Sea survival thing was that getting out into the offshore at the right time is critical to survival. And that's sort of the critical period. And what really happened after studied a bunch of different river systems and a bunch of sites is that it didn't pan out the way the initial study was. That early marine growth is important regardless of the habitats. And in the case of the Nooksack, you had kind of steady growth through all the different habitat types. This is standardized growth, not absolute growth. Small fish grow slower than big fish, but to that small fish, it's the relative growth that matters. So I'll blow through a few pictures here. So top 2 different years. So I have water temperatures. A lot of that growth is dependent. They're cold-blooded creatures, so growth is dependent on an optimal temperature. I blocked it out. It's sort of 9 to 16 degrees is sort of the optimal Chinook growth. And then here's how many fish are in the system, the different places, and here's what their growth is. So in one case, in 2014, it was good to be in the pocket estuary early, and then later the delta and pocket estuaries were about the same growth. And then when you got to the offshore, it was either similar or bumped up a little bit. In 2015, a different year, different temperatures, but the pocket estuary wasn't as good as being in the delta, and then when you went out into the offshore, it was pretty similar growth. So does it any of it matter? Because like Jeff just showed us that somebody eats them anyway. So this, and Michael showed this too, and this is a really important study that Lance Campbell did, because he shows in this contemporary time period that we have, with all the bad things happening in the Salish Sea and on land, we have fish that are surviving, and we can trace them back to what life history types they are. He does that with otolith chemistry. Basically, they can run a laser on the otolith and measure calcium or strontium calcium ratio, which correlates to salinities. So you can figure out an ocean entry period. They call it ocean entry, but it's when it basically is 4 parts per thousand salinity. So in this case, the fish entered that salinity at about a 42 millimeter size fish. That was a fry. In this case, it was a par. So, and Michael showed this slide, and basically, in the North Sound, the small fish are surviving. These are the box plots of the fish that survived. Returns in 2015. If you remember an earlier talk, 2015 was a really bad return year, at least for South Fork springers. So there's probably no South Fork springers in that. There were 7 that returned, I think they shed. But small fish are returning to the spawning grounds. And in this case, it was, if this his sample of fish represent the population in the Nooksack, it's 28% of those were fry. So those are fish that encountered the estuary basically just past just downstream of the tidal area, so scrub shrub estuarine zone, and where all that phenotypic branching occurs, those are fish that are surviving. It's included in that population, and you have par migrants and you certainly have yearlings. So that's basically it. So what does it all mean? You have the fish that are expressing the life history types here. There's a lot of restoration potential. And kind of the bottom line that I want to say is, like, let's not get too worried about current conditions. Think about the habitats that you need for the desired future condition of the population. And I know that, well, now there's 190,000 fish out migrating. The recovery plan, unless it's been updated, says they want to have about 1 million out migrants. So you're going to need more habitat. You're going to have more fry. And so that's it. And probably there's no time for questions.
Thank you, Matt.